Good afternoon, everyone. Thank you very much for joining us for our Cambrex Insights briefing today. So our discussion for today is really focused around navigating the solid form landscape during API development. So what we're going to do today is I will give you a brief introduction to Cambrex, just an overview of who we are and what we do. And then we'll move on to look at the solid form science of APIs in a lot more detail. So we'll look at how understanding the solid form landscape of an API can help with drug development and help us to be able to minimize the risks during both drug substance and drug product development. So we'll go through a case study which really focuses on hydrates and really how to navigate around undesired forms when we're looking at both anhydrous and hydrated materials. So just to give you a bit of an introduction to Cambrex. So we are a CDMO. We have around 2,000 employees, and we're really focused on small molecules. So our focus across all of our 12 facilities is on small molecule development. As I mentioned, so we have 12 facilities globally. This map here just gives you an indication of where those facilities are based. So we have 5 facilities in Europe, and then also 7 in the United States. And all of these facilities are really focused on both drug substance and drug product development, as well as analytical development. So within these facilities, so what we have, we are really able to support both drug substance and drug products right from preclinical development all the way to commercial. So we have different sites that have the capabilities to scale up to different volumes, different levels. So for example, 200 liters, 2,000 liters, and then all the way up to commercial manufacturing for drug substance. And then our drug product facilities are also able to take your API right from preclinical all the way to commercial. At these sites as well, we also have many analytical development groups who are able to support with method development, as well as things like stability studies as well. So a little bit closer to our topic for today, we also have a site in Edinburgh, so the Cambrex Edinburgh site, which has a team of solid form scientists who are really dedicated to looking at what is the most suitable solid form for further development. So we look at which solid form should be chosen when moving forward with your API, and also how we go about scaling up that solid form for manufacturing. So this team is particularly focused on supporting the drug product and the drug substance groups in terms of ensuring that we can de-risk the solid form for these other sites at Cambrex. So just to move on to our talk for today, so really we're going to be looking at polymorphism in a lot more detail. So a polymorphic form, an API exhibits polymorphism when it has more than one crystalline phase of a particular compound. So when there's more than one crystalline arrangement of your molecule for a compound, that's when we say that the API is exhibiting polymorphism. So we often get the question from our clients on why should they actually look at polymorphism, how much effort they should put in to looking at the polymorphic landscape of an API. And really, we only need to look at the properties that differ between polymorphs to understand that actually the polymorphic form affects pretty much every step of API development. So we've listed here some of the properties that the polymorphic form would affect. So for example, physical and chemical stability. If we have 2 anhydrous forms, we might find that one form is physically more stable than the other, or chemically more stable than the other form. Hygroscopicity. We may find that if we leave one form on the bench exposed to humidity conditions, it will pick up more water than the other form. Things like bioavailability and also solubility. So what we see there is that 2 different polymorphic forms might exhibit different properties with regard to bioavailability, different dissolution profiles. And when that's the case, it can actually have a lot of effect during your clinical studies. So polymorphic form is critical when it comes to looking at solubility and also bioavailability. There's also other aspects such as your drug substance manufacturing and your formulation, where the different polymorphic forms might have different crystal morphologies. So they might have maybe a rod-like particle versus a needle-like particle. And this will change the handling properties of the APIs. So things like formulation, filling of capsules, it will be very different for one polymorph versus another if they have very different crystal morphologies. So we need to take all of these factors into account when looking at what polymorphic form we choose for further development. So essentially the polymorphic form becomes critical at every step of the drug development process. And this is really why we spend a lot of our time trying to take an in-depth look at the polymorphic landscape of an API to really understand what are the risks associated with the form that is chosen for further development and what other forms might be out there that need to be taken into account when scaling up and when doing both drug substance and drug product manufacturing. So when we carry out our polymorph screening studies, what we normally do is a very extensive polymorph screen, which basically involves running about 150 experiments, looking at different solvent systems, different crystallization conditions, in order to understand what solid form is coming out of each of those conditions. Now, ideally, you would count yourself a little bit unlucky if your polymorphic landscape looks like this, because this is pretty complex. So most of the compounds that we work with, you won't get an as complex polymorphic landscape as we see here. But if you do, really the critical thing is to understand what conditions each polymorphic form is generated from. So it might be a cooling crystallization, anti-solvent addition crystallization, a particular solvent system that leads to the polymorphic form being obtained. But the key thing is to really understand where those forms are favored. And that's really why we carry out our polymorph screening work, is to gain an understanding of what forms are out there and which ones we should be moving forward with into further development. So one of our focuses for today will be on hydrate formation. Now, there's different ways of classifying polymorphs. So scientifically, polymorphs normally have a thermodynamic relationship to each other. But there is another class that we often come across during our polymorph screening work, and that is solvates. So with a solvate, you will have normally an organic solvent molecule incorporated into the crystal lattice for your API. And normally solvates are not developable, they are not developed, but we do need to know about them. So we need to know what solvent systems give these different solvates and which ones to avoid when we are doing manufacturing. But there is a special case of solvates where it's not an organic solvent that we're using, but it's actually water. So this is where we get one or more water molecules incorporated into the crystal lattice to form a hydrated material. So it could be a monohydrate, a dihydrate, or even a higher hydrate, really just in the presence of water. So hydrates are one special case of solvates, but they're really important for us to look into them and understand at what point they are actually formed. It's estimated that about 75% of all pharmaceutical compounds have the ability to form hydrates. So it is a big concern, particularly if the hydrate is not the solid form that is being developed. So we really need to look into our API landscape to work out if hydrate formation is possible. Normally we find that your API will be exposed to water at some point, so actually various steps during the development cycle. So for example, during drug substance manufacturing, we might have water in some of the reagents. During formulation development, wet granulation, lyophilization, even present in some of the excipients, we may have water there. So it's really key that we understand what the effect of water will be on the solid form. So is it actually going to form a hydrate? Is our API, if we choose an anhydrous form, is that form likely to convert over to a hydrated material over time or under one of these conditions? Or are we safe? Do we have a good, robust anhydrous form that we're looking at? Another key thing is that when the API or the drug is actually administered, there's also the chance of hydrate formation at that point. So we really need to understand whether it is a risk to your API or not. So that really leads us into our case study for today. So in terms of our case study, what we're going to look at is how we actually go about understanding what polymorphs an API actually exhibits, and then how we navigate around the unwanted polymorphs that might be present for that API. And we'll look at hydrates in particular. So just to give you some background for this case study, so we worked on an HBr salt, so hydrobromide salt was the API here. We received this compound. We never had much information on it. There was very little information received regarding the solid form. And the aims for us were really to look at initially which is the best solid form to move forward with into further development. So which is the most appropriate solid form to select for scale up. And in order to do that, we carried out a polymorph screening study initially. Once we had carried out the polymorph screen and we had identified the different solid forms that might be present, we started to look at the risk of hydrates. So we wanted to see whether we get hydrate formation for that compound, and also where the hydrates are formed, so under what water activity the hydrates are actually obtained. So for that part of the study, we were looking to establish the operating range for the anhydrous form versus any hydrated forms that were observed during the polymorph studies. We then moved on to look at developing a robust crystallization process for the scale-up of the solid form that had been chosen. So we have a look at what are the different crystallization process conditions that will allow us to be able to scale up our chosen solid form. So this gives you an example of the type of readout that we get from a polymorph screen. So the way we carry out these types of studies, these types of screens, is to look at initially around about 24 different solvent systems in about 5 different crystallization conditions. So what we're doing here is exposing the API to different conditions that will favor different polymorphic forms being obtained. And what we saw from this screen is that you can see a lot of ones there. So normally the form that dominates throughout the screen is normally the thermodynamically most stable form. Not always, but a lot of the time it will be. And then we also have a lot of other colors there. So we identified 9 different forms, 9 different solid forms during this polymorph screen. A lot of these ended up being solvated materials. So form 2, 3, 4, 5, and 6. These were all solvated, and you can see that they come mainly from the one solvent system, each of them. But then we also identified 2 forms that actually came from conditions where there were higher water percentages present. So form 7 and 8, so the red one there and also the blue one, those ones were obtained when we had either water on its own or a higher percentage of water present within the solvent systems. So that immediately tells us that there is a risk of obtaining a hydrated material, just at least initially from this polymorph screening study. We obtained 9 forms, so I know we have 8 listed there, but what we did see is that form 8 actually dehydrates to an anhydrous form 9 material when it is taken out of water or out of the solvent matrix. So that's why we had 9 forms coming from this particular polymorph screen. So what we do once we've carried out the initial polymorph screening work is to look at what are the solid form properties of the forms of interest. So form 1 was the obvious one to look at. That came out in a lot of the experiments during the polymorph screen. And then form 7 and 8, those are the ones that look to be the ones of higher risk. So we need to look at those in a bit more detail from a characterization perspective. So what we're looking for when we do the characterization is to identify exactly what the solid form is. So is it anhydrous, or are we seeing a monohydrate or a dihydrate coming out from the polymorph screening studies? We're also looking at crystallinity. So we do XRPD analysis to look at how crystalline the material is, how difficult it might be to crystallize moving forward. We look at the morphology of the crystals. So are we getting a needle-like morphology? Are we getting rods or blocks or plates? Because that will all impact further steps during development. We look at the melting point, so the DSC analysis to look at the melting point And we also look at things like hygroscopicity to see how much water the API will pick up under elevated humidity conditions. So there's various studies or various tests that we do here in order to really get a much better understanding of the solid forms. Both Form 1 we characterized here, and then also Forms 7 and 8. So overall, Form 1 was a really good solid form. It showed a high melting point. The hygroscopicity was very low. There was a challenge with regard to the morphology. There were a needle-like morphology, which we addressed later on. But Form 1 looked like a very good anhydrous form to move forward with into development. Form 7 we characterized as being a dihydrate, so 2 molecules of water within the crystal lattice. And what we find for Form 7, and the DVS profile is down at the bottom here, is that there's very much a stepwise loss of water. So there's a stepwise increase and a stepwise decrease in the DVS analysis, which is very common for hydrates. Often you will see that there's a stepwise change in the mass when we run DVS analysis for hydrated materials. And what we see here is that below 30% humidity, the hydrate starts to dehydrate. And at this point, we started to see it becoming amorphous. But the good thing is that we did now understand what the properties were for Form 7. Form 8 was less of a worry. It was not stable, so when taken out of solution, it was not stable. It turned to Form 9, which was another anhydrous form, but less stable than Form 1. So at this point, we had a good understanding of the 3 forms of interest for the API. So although Form 1 looked like the best solid form for further development, it did have its challenges that we needed to address. And one of those was really the fact that it could actually form hydrates. So we could get Form 7 and we could get Form 8. This is a snapshot from the polymorph screen indicating the higher water percentage and higher water activity solvents that gave hydrated materials. Now, the one option for manufacturing and scale-up would just be to avoid solvents that have water, so to choose solvents that weren't holding on to much water. But the problem that we have here is that we were forming a hydrobromide salt, and so the actual hydrobromic acid was going in as an aqueous solution. So that was one of the challenges where we're actually introducing water into the system during the salt formation, and so we needed to understand under what water activity conditions these hydrates could have been formed. The other challenge for the Form 1 material was that there was very limited solubility in organic solvents, but there was high solubility in water, which meant that there was not much choice with regard to process solvents, so at least process solvents that were not holding on to too much water. So we set about to look at how much water could be in the system in order to be able to still get Form 1, but navigate around the hydrated forms. There was a further challenge with Form 1 as well that we addressed later on, and that was the particle morphology. You can see in the microscopy image there that we're getting very small particles, needle-like particles. The density was very poor, poor flowability, and very difficult to handle. It also filtered very slowly as well. So from a crystal perspective, a solid handling perspective, it wasn't looking great, at least from the initial screening work that was carried out. So really there were a few different challenges that we needed to look into for this Form 1 material. So to address the first one, what we did was look into a hydration mapping study. So this is where we really try and map out under what water activity the material will change from being an anhydrous form to a hydrated material. So we go about doing this by looking at the water activity over a particular sort of percentage of water. So for example, we go for water activities between 0.1 and 1, and then we also look at the corresponding percentages of water for the different solvent systems. So we went for 3 different solvent systems here. So these were all potential solvent systems for the crystallization process. So we looked at acetone with mixtures of water. We looked at ethanol with mixtures of water, and then also 2-propanol as well. And just in terms of some of the properties of water activity, you can see that the water activity curves are not linear. So the water activity, it will change as the water percentage changes, but it's not a linear change, and every solvent will have a different water activity profile. It's also solvent and temperature dependent. So we really need to look in some detail at how our solid form is going to change in these different solvent systems, and what is the critical water activity for the crossover between Form 1 and Form 7 and 8. So we carried out a hydration mapping study, and this was really the readout of that study. So what we see in there, and the way this is set up, is to create slurries of the API in different solvent water mixtures that equate to different water activities, and then look at the XRPD analysis of the solids to work out what form is favored at a particular water activity and a particular temperature. The choice of temperature is normally dependent on the crystallization conditions, so we tend to go for temperatures that cover the crystallization range, so between 5 and 50 degrees. It would be our normal selection there for the temperatures that we look at. And really we're looking to establish the boundary points, and what we saw here is that the critical water activity for this API at 5 degrees was between 0.4 and 0.5. So that's the point at which the Form 1 converts over to Form 7. For ethanol water, it was a little bit higher, but they're all within that same region. In terms of the ambient temperature results, we see that the critical water activity is between 0.5 and 0.6, and then at 50 degrees, it's around 0.7 there. So the water activity, or the critical water activity, will change as the temperature changes, and it's obviously also dependent on the percentage of water there. But this data really allowed us to establish what are the operating ranges for obtaining Form 1 versus getting Form 7. And you can see that Form 8 is at the bottom there. So Form 8 is obtained at very high water activity levels, but that was less of a concern for us. So in terms of solvent selection, we went on to choose ethanol water as the process solvent for this crystallization. And that was really based on a few things. So ethanol water gave us the best chance of having a fairly significant amount of water present in the system without actually forming the hydrated material. So you can see there that we can have up to around 8.9% water in the solvent system, and we will still maintain Form 1. So that was one of the big reasons for choosing ethanol water over the acetone or 2-propanol systems. We also looked at solubility data. So the numbers that you see in this table here, this is the solubility data that was generated at each of the different water activities and temperatures. And then we also carried out metastable zone width measurements to see how broad or narrow the metastable zone width was under these conditions. And overall, based on the data that was obtained, we selected the ethanol water system as being the most promising solvent system for this crystallization. Now, what we went on to do next, so at this point we start to look at what is the best crystallization pathway. So once a solvent has been selected, what is the best crystallization pathway to choose? And there's a lot of different options. So that is our operating range. So within the orange space there, that is the operating range for our crystallization for obtaining Form 1. We then can explore different pathways. So for example, we can start at around 120 mg per mole at the elevated temperature, cool down, and then add some anti-solvent in order to boost the yield. That's one thing that we did look at. Or we can start to add anti-solvent first, so add anti-solvent at a higher temperature and then cool in order to reach an endpoint for the crystallization. So that's another pathway that we could look at. We could also start at a lower temperature. So for example, starting at 50 degrees, adding anti-solvent and then cooling as well. And then there's basically an intermediate type process where we do some cooling, add anti-solvent, and then cool further in order to get down to a low temperature and also ensure that we get a good yield there. So there's various different pathways that we can take, and we carried out a lot of smaller scale crystallization trials in order to see what the best pathway was for this crystallization. So this was very much vial scale initially at this point. So based on the data that is generated on the small scale, we then move on to do our process development in controlled lab reactors. So we used our automated reactors where we have basically a blaze metrics probe, which allows us to be able to look at nucleation. It gives us real-time data on the nucleation points, on any crystal growth. It gives us online microscopy. So essentially what we're able to do is see at every point within your crystallization vessel, are the crystals growing? Is there agglomeration? Is there oiling out? That's the type of data that the probe allows us to gather. And so we look at when we're doing the crystallization development, we start to look at what are the critical crystallization process parameters for this crystallization. So we look at the cooling profiles, at what rates we should be cooling at, should it be linear cooling or not. We look at anti-solvent addition. How quickly should we be adding the ethanol in order to get to the end point, and also at what temperature the ethanol should be added. We look at hold periods. So if we hold the crystallization for a certain amount of time, are we getting more material coming out of solution, or does it reach a steady state where we're actually not getting any changes, or are the particles breaking during that hold period? Because we're operating at elevated temperatures, we also look at solution stability. So if we were to hold our crystallization mixture at an elevated temperature for an extended period, what would happen to that mixture? Would it be stable? And then we also go on to look at things like filtration and drying and washing in a lot more detail as well. So the crystallization scale-up development is really focusing on developing a process that can be used for further manufacturing. So over the next few slides, we'll go through some of the data that is generated from these crystallization development work programs. So we do carry out a number of experiments, at least 8 to 10 crystallizations on the larger scale. And we'll go through the results of 3 of these crystallizations. So the first crystallization there, that was basically seeded, and then we've cooled down to 5 degrees before adding anti-solvents. And what we see in there is actually the particles for that one are pretty poor. So we're seeing some agglomeration, we're seeing breakage of particles, very, very small particles as well. Definitely not an ideal process. The blaze metrics data, again, is showing us the trends that we see with regard to the number of particles for each particle size level. So anything below 10 microns or between 10 and 40 microns, all the way up to 150 to 1,000 microns. The blaze metrics data can give us that information on which particle size which is actually growing during the course of the crystallization. So are we getting more fines coming in, or are our crystals growing larger? And for this one in particular, the first one there, the particle size is very small, and it's really quite poor. So from that, it looked like cooling first was not the best option, and very seldom is, actually. So what we went on to do then is a second process where we have added anti-solvent at 60 degrees, so slowly added ethanol at 60 degrees before cooling down to 5 degrees. And what we see here is a much steeper curve in the blaze metrics data. So this essentially means that the material is crashing out of solution. It's coming out very quickly during the ethanol addition. And it's definitely not controlled. So from this data, we get an idea of how controlled the process is, and it wasn't controlled at all. And you can really see it from the particles on the bottom there, that we're getting different size crystals, and a very elongated rod-like particle was obtained from this crystallization. So definitely not ideal and not controlled at all. This basically is a further data that we get. So from that prior crystallization on the previous slide, this gives you the online microscopy information that is generated from that. So you can see that the blaze metrics probe, it gives us an indication of what is happening within the reactor. So are we getting rod-like particles? Are we getting breakage on the crystals? And also what the approximate particle size is as the crystallization proceeds. So this is the type of data that we need to collect in order to better understand the crystallization process. So the most promising crystallization process that was obtained was this one here. So what we did for this one was seeded at 60 degrees, so cooled down from 70 to 60, seeded, and then what we've done then is next cool down to 50 degrees and slowly added ethanol. The key thing here was to add ethanol quite slowly so that we got better control over the growth of the crystals, and also lower aspect ratio crystal as well. And then after the ethanol addition, we've cooled down to 5 degrees. But really, you can see from the Blaze metrics trends there that it's more gradual, so you get an increase in particle counts more gradually, and that's a good indication that we are getting control over the process. So this one ended up being best. You can see the difference between the crystals that were obtained here versus the ones that we had in earlier slides, which were very much more sort of needle-like, smaller particles, difficult to filter and handle as well. So these are the types of crystals that we were aiming for during the course of this work program, to really have a look at how we can control the crystallization and get crystals that are of a better quality for further processing. So it's all very well as developing the crystallization process in the lab. In the lab, we are working at about 100 ml scale. We can go up to around 5 liters at our Cambrex Edinburgh facility. But ultimately, the crystallization process needs to be transferred to a manufacturing site for further scale-up and drug substance manufacturing. So what we would normally do during these work programs, and what we did here for this case study, is go from 100 mils up to 1 liter initially, and then we took it up to 5 liters just to demonstrate that initial scalability for the process. And really the way that we do this is through Dynachem modeling. So we use this modeling software to be able to do a scale up and scale down model. What we would do is input the parameters for the reactor into Dynachem for both the smaller scale reactor as well as the larger scale, and allows us to be able to see what kind of mixing rates we should be using at the plant scale in order to ensure that we get consistency in the way that the crystallization proceeds. So we carried out this Dynachem modeling study, and then we were able to scale up firstly to the 5 liter reactor within our laboratories, and then transfer over to the manufacturing facility and provide this information to them on what kind of stirring rate should be used. And the process definitely worked very well. So the transferability was really good. And we find that it is very effective. The modeling tool is very effective in allowing us to be able to get a representative process on the small scale that can then be scaled up during manufacturing. So from this process, we were able to get a manufacturing procedure for the crystallization that favored form 1. So we never saw any of the hydrates at any point during the further crystallization development, because we were operating in the favored form 1 zone or operating space. We also significantly improved the particle size and the crystal morphology. So this allowed for better filtration rates as well as handling of the actual crystalline material. We had effective impurity purging. The crystals, because they were very discrete, they weren't small, it allowed for better impurity purging. A good yield, so we got over 90% in terms of yields. I think the process volumes were around 14 for this process, so reasonable there. And we were also able to isolate the crystalline material very effectively. The filtration was very fast, the drying was also effective, and it also helped with the handling properties. So in terms of achieving the goal for the crystallization, this process and the development work that was carried out definitely allowed us to be able to deliver a much better material, a much better solid form after carrying out the crystallization development. So just really in summary there, so really what we see is that carrying out a polymorph screen is critical in order to really understand the landscape of your API. It's also a regulatory requirement, so polymorph screening is a regulatory requirement, and it has to be carried out before the launch of a particular product. We also need to pay particular attention to hydrate formation. As we've seen during the case study, it's important for us to understand the hydration space of the molecule, and also what are the critical water activity levels. Solvent selection is also important. We could have chosen acetone, but I think we would have had a lot more problems if we had assessed the acetone system. We want to choose a solvent system that gives us the broadest operating range for our crystallization. And really, I think what we see from the case study there is by developing a controlled crystallization process, it allows us to improve the solid forms. And if we can improve the solid form that has been developed, it basically cuts down the time of development, it speeds up the development, and essentially helps our clients to be able to get to the clinic a lot faster because it avoids problems like obtaining the wrong polymorphic form during scale-up or obtaining a hydrated material when it was not required or when it was not desired. So I think hopefully what we've gained from the study is that we can really see that understanding the solid form landscape and navigating around unfavorable forms really helps us to be able to reduce the risk of obtaining the wrong form during both drug substance manufacturing and also during drug product manufacturing. All right, thank you very much. Welcome. Any questions?